Every day, billions of cells die and are then cleared, a process necessary for homeostasis and an organism's health. Although there are dozens of cell death programs, the three most characterized forms of cell death are apoptosis, autophagic cell death, and necrosis. Once dead, cells are cleared by either professional or non-professional phagocytes. The McCall lab studies specific proteins that may be responsible for cell death and clearance, such as Draper and SED12. Recently, they found that kinases of the J&K signaling pathway are required in the phagocytic follicle cells to eliminate germline material in fruit flies. As kinases are important regulatory enzymes that have been implicated in many diseases, including cancer and neurodegeneration. Our study focuses on identifying other kinases that may play a role. This research was completed using the Drosophila melanogaster, which provide many advantages in studying genetics as they have a simplified genome, fast generation times, and similar kinase pathways as humans. Drosophila egg chambers are composed of a germline of 15 nerve cells and an oocyte surrounded by a layer of somatic follicle cells. The egg chambers in the ovary develop through 14 stages of oogenesis, during which the nerve cells provide nutrients for the oocyte. The nerve cells are then eliminated by the follicle cells before the oocyte is laid. This naturally occurring developmental death allows us to study pathways involved in the clearance by follicle cells. When critical pathways are disrupted, a number of defects may occur. An example of this is when follicle cells fail to clear away dying nerve cells. In this experiment, we purposefully disrupted these pathways in order to determine if certain kinases played a role in clearance. We screened 11 kinases to test for corpse clearance defects related to developmental death. To test the effects of each kinase, we knocked them down using the Gal4-UAS system. In our experiment, the follicle cell enhancer GR1 enabled the Gal4 protein to be expressed in the follicle cells. The Gal4 protein then activated the UAS, an upstream response element that activates a gene of interest, in our case, the kinase RNAi. The kinase RNAi is a double-stranded RNA and is therefore eliminated by the fly's naturally occurring enzymes, causing a knockdown of the kinase. Knocking down crucial kinases led to defects in the clearance of nerve cells by the follicle cells. The resulting flies were dissected and their ovaries were stained with DAPI, which highlights the nuclei of cells and can be used to see death and persisting nuclei. Nuclei that have not been engulfed due to defects. Once defects were found in relation to developmental death, lysotracker staining, which highlights acidification in nerve cells, was used to observe other defects. The kinase mutants of center divider AMPK alpha, CG8298, monopolar spindle 1, CG11486, and CG7766 were all found to have persisting nuclei during developmental death. Nerve cell nuclei in CDI, AMPK alpha, and CG8298 knockdown flies demonstrated more acidification than flies with RNA against Draper, a well-characterized engulfment gene. These findings will allow researchers to gain a broader understanding of cell death and corpse clearance, which play roles in diseases such as cancer and neurodegeneration. Thank you.